Back to you.
Thank you very much. Okay, so we're now going to move on to our grant, which is looking at the formulation for 3D printing. So creating a plug and play platform, disruptive UK industry. So hopefully we'll very shortly be linking on to Ricky. So to hear sort of how the project has been addressing the research challenges and formulation for 3D printing. So Ricky, are you able to share your slide?
I am just selecting my slides now.
Brilliant.
Okay.
Fabulous. Yeah, yeah.
It's good. Okay, so
just open, yeah, as a full presentation.
Let me just put it into presentation mode.
Yeah.
And, slideshow. Okay, thanks, Helen. So I'm Ricky Wildman from University of Nottingham, and I'm going to talk about our project, Formulation for 3D Printing. And this, of course, was funded by EPSRC as part of the Future Formulation endeavour. And this project is supported by a number of companies across a number of sectors, and they're all listed there: GSK, PPG Incorporated, which is a mainly US company, Syngenta, and Malvern Panalytical. So our project really started with a problem, and this problem was really thought about by two of our colleagues, Tim Minshall there on the left, and Phil Dickens on the right, and they received some funding to be able to go around to a number of companies and say, we've all heard about 3D printing, additive manufacturing, or why aren't you using it? And so they created this national strategy for 3D printing, AM National Strategy for the UK, and went around these companies and really tried to find out what was going on, what was stopping companies from adopting this as a full-on production process. And a number of reasons came out, things like reliability or the scale or the throughput of manufacturing. But it turned out that the number one problem that companies in the UK identified was the lack of materials, and specifically the lack of materials available to them for their specific applications or products. So what we decided that we would do is we would really try to tackle this. We would try to identify some materials that these sectors, across sector collaborators would be interested or would want to use in the development of their products, but also find ways in which we could find materials much more quickly than we had done in the past, and then go on to try and exploit all these kinds of levers for use of additive manufacturing in industry. So we were funded, fortunately, evidently. We got a multi-institutional project, so there's Nottingham, Birmingham, and Reading, and supported by some really key and very supportive industrial partners, which I've mentioned before. And so we set out with this project to really try and see whether we could engage with these industrial partners to really understand what the problems in terms of materials were, and then try to find ways in which we could discover materials much quicker, because it was very clear that the materials weren't available. They weren't on the shelf, and we couldn't just pull them off the shelf, and we would have to go out and find them ourselves. Now, in the course of that, we developed the means by which we can more rapidly identify materials suitable for 3D printing, and I talked about that in previous future formulation meetings, but I'll touch on it in this presentation. But more than that, over the course of these 4 years, we've really explored various different ways in which, once we found the materials, we can exploit the material properties and the integration with additive manufacturing to be able to exert greater control over products than perhaps you might be able to with other manufacturing techniques. And so we thought of them as levers, various different levers that would allow us to formulate for 3D printing. So in the course of this presentation, I'm going to go through a series of these. Some of them are quite obvious, but as we go on, hopefully they'll become much more, I guess, complex and less obvious, and actually much more towards formulation, as most people in the formulation field will come to understand it. So various things that we found that we can do, some of them which we already knew and are obvious. So for example, if we want to change the performance of a product, then we can simply reformulate the material. We just change the material composition or change the components and then produce a new product. And I'll show an example of that later on. But here we have two different types of tablets for two different types of APIs, drugs, where we had to reformulate the material to get different properties. Beyond that, we took a screening approach. Instead of taking a hypothesis-driven approach to identifying materials, and by that I mean we would say this is the need, and then this is the type of material that would suit that need, and we would explore iteratively to try and prove this hypothesis. We would sort of take the analogy of throwing things at the wall and see what would stick, and really take a screening approach, trying to tackle as many materials as we could in one go and just try to slim that down to find the materials that were really working out. Of course, as most people know, if they're familiar with 3D printing, additive manufacturing, you can change shape. That's one of the attractive parts of doing 3D printing. You can change the shape and you can change the properties. And that's the same for whether you're trying to release drugs or release agrochemicals, or you're trying to change the structural performance. You change the shape and you get some performance difference. And I'll show you some of the things we've done with that. And then we can start to think about microstructure. We've seen a little bit about microstructure in other applications in these talks, but we start to think about how we can understand the microstructure that arises out of additive manufacturing that perhaps wouldn't arise in other technologies, and then how we can exert control through our choice of materials and how we blend them together. And then finally, instead of blending, we can think about instead of mixing, we can, with 3D printing, we can print more than one material at a time with some really quite exquisite control. And so we can actually form that microstructure by design rather than allowing, say, the kinetics to play out. So the first thing is, okay, we can reformulate, and we can really think about how we can reformulate the material composition of a product to be able to get different properties. And one of those, a good example of that is when we came to try and 3D print tablets, solid dosage forms. So these are the tablets that we're all familiar with. They have a drug encapsulated within them. Often they're created with tableting, sometimes in capsules and so on. And working with our partner GSK, we wanted to see if we can try to find some common thread of materials or composition that would allow us to have a kind of a general ink for 3D printing these kinds of tablets. And many people in the audience, of course, know about pharmaceuticals probably much more than me. And these drugs have various different properties, and a common way of classifying them is in their solubility or their bioavailability. And so we tried to tackle a drug that was on the more soluble end of an API, and ones that were on the more poorly soluble end of drugs, and see if we can find a formulation with common components to enable us to be able to get them into the tablet in the first place, 3D printed tablet, and then allow them to release. So the first one was relatively simple, I would say, though it took a long time for us to be able to formulate this, and led us directly to the idea of going to high throughput methods. But if we think about ropinirole, it's a highly soluble API, it's a highly soluble drug. And we decided to go along a kind of a hydrogel type approach to being able to encapsulate that into a printable form. So we created a formulation around polyethylene glycol diacrylate, commonly used as a cross-linker, and we used that in combination with a photoinitiator for UV curing our drug, and water. So the water is there for essentially two roles: to be able to get the drug into the tablet in the first place, and then of course then it allows it to be released once it's ingested. And we were able to print this successfully. We were able to cure it. There were tablets that you could hold. It released as per the criteria set out by our partner company, and it was a stable drug in this form as well. So we had a really nice formulation. It met everything that we wanted out of a formulation that could lead to 3D printed tablets. So when we came to then thinking about poorly soluble APIs, we then have a pretty difficult problem because we then have a lot of water in there. The water's been used to dissolve the soluble API. When we've got a poorly soluble API, that's not going to go in at all. We're going to have a bit of a problem. So we decided to try and keep the core of the UV curable element of our formulation, the polyethylene glycol diacrylate, the ErgoCure, which is the photoinitiator, and then we've got the carbidolol in there, which is poorly soluble. But instead of the water, we chose to go to a different kind of solvent. So we swapped out the solvent. We modified the proportion of the solvent in there compared to the water-soluble, and then we were able to then print that. We were able to get the drug in, we were able to get into a nice formulation, we were able to print that. And on the left, you can see examples of where we were able to print, and we were able to print in various different formulations. So the MVP is a really nice choice because when you cure it, when we cross-link it in with the PEGDA, when we link it into the PEGDA, it creates PVP, which is a common pharmaceutical excipient. And so as long as you can get the MVP out, or you can fully convert it, you have a nice form of a tablet in here. And as you can see from the release studies, that's the cumulative release, percentage of the release against the time. We can see we can get a reasonable release of that drug from that formulation. And we're able to print it in various different forms. So with this, we're able to show that you do have to reformulate. You reformulate to get different properties and different release, but you can at least get a sort of a backbone of a formulation that allows you to be able to change your formulation in a small way to get different kinds of drugs out of your formulation. So next, we wanted to really think about what you do when you don't really know what you're doing. And in this case, we wanted to really think about other function instead of drug release. We thought we had a nice handle on drug release. We know how to formulate these things for other manufacturing techniques, but where you don't really know what you're looking for in terms of molecular structure and how it's going to behave, you have a tricky problem. And so what we decided to do was buddy up with some colleagues who were creating screens of polymers, and typically UV curable polymers, and looking for various different, what we call cell-instructive behaviors, and in this case, resistance to bacterial attachment. So what we decided to do was key into that screen, identify polymers that look like they would be printable, and then scale them up and demonstrate that when you 3D print them, that you could take it all the way to the application stage. In this case, in vivo preclinical studies, and we were able to do that. These are the sort of examples of the kind of monomers that we were faced with. Our colleagues screened those, identified them in a high throughput manner, which of those had the cell-instructive properties, and then simply by looking at the kinds of viscosities that these monomers had, we were able to make a reasonable guess about which of those monomers would be printable in an inkjet printer. So we down-selected two monomers. They're essentially a monofunctional or difunctional a pairing of monomers that were shown to be resistant to bacterial attachment. And so we then scaled that up, printed samples, put bugs on them, and basically saw whether the bugs liked to stick to them or whether they didn't like to stick to them. And we were able to see that compared to commonly used materials, so PDMS, polysiloxane silicone, and materials that we knew that would promote attachment, our formulations showed a really nice resistance to formulation. And essentially, these four formulations are variants in the amount of photoinitiators we had in. And we saw that the better curing you got, the better cell-instructive resistance to bacterial attachment that you got. So we were really happy. So we were able to take from a screen, we would be able to identify materials, and then we were able to show that you could print them and that they retained functionality, at least in vitro. And then we went to the next stage, which was to do in vivo studies. So we constructed an exemplar, we made samples ready for creating in vivo studies, and then we tested those within a mouse model. And we saw that our formulations, despite having a really hefty dose of bacterial infection inoculation, cleared out the infection in vivo really well compared, 3 orders of magnitude compared to medical grade silicones. So we're really happy with that. It really shows a lot of promise for 3D printed formulations, but also it shows the promise of taking a screening approach to identify material properties. We also did this for antifungal materials, so we took a similar approach. In this case, just stopping it in vitro, but again, in the bottom right, you can see 3D printed versions of materials that have been identified in a screening approach to resist fungal attachment, candida in this case. And when we tested our 3D printed constructs, the most interesting it really is the one on the bottom right, which is essentially a voice prosthetic, commonly again made out of silicone, really subject to prevalent fungal infection when it's placed in the body, and we showed that we can manufacture fungally resistant, personalizable 3D printed constructs. So that was really nice for us. We were able to show that you can screen for various different cell-instructive properties, fungal resistance, microbial resistance in the wider thought of it. So the one thing that we didn't do, the thing I mentioned there, is that basically we eyeballed whether we thought it would be 3D printable or not. We eyeballed and said, this is the viscosity. We kind of know what the viscosity is, but that's not really a high throughput method. And so the one thing that we did then to augment this kind of high throughput screening approach for materials is to then bolt on a high throughput identification of printability, specifically in this case for inkjet, but we have variants for other 3D printing systems as well. And so our system was a very simple system, really. We used a liquid handler to automatically aspirate and dispense target liquids, target monomers, and then using a pressure balance, pressure drop balance, a force balance to look at the rate, the pressure over time for a given flow rate of material, and extract out of that what the viscosity must be, and then calibrated that against known materials. Then for surface tension, which is also a driver or an indicator for printability, we did something very similar for a pendant drop, performed a force balance on that, and automatically measured the force balance with our liquid handler. So without going into too much details, we can see that basically, or we know that viscosity goes down over time. We tested a whole bunch of materials on the right, and you can see viscosity goes down with temperature. And so just as a quick test, we took a whole bunch of materials that really weren't printable at a low temperature, room temperature, and we increase the temperature, the viscosity comes down, and you can see that the printability gets better and better as we increase the temperature in line with what we would expect. And then we did our sort of litmus test. We took a material that we predicted to be not printable at 50 degrees. That's the material that's labeled on this screen, and the same material would be printable at 60 degrees. And we tested that on our printers. And so on the right, you can see up at the top, this black blob at the top is the nozzle, the print head nozzle, or a print head nozzle. And you can see in the upper scheme, there's nothing coming out of this nozzle. It's a no-no. It's not printable. And then in the bottom row, we can see a thread of material that comes out, showing that it's ejectable, it's printable. And so this was our litmus test and showed that our method, our screening method, was predictable and a good guide to whether something would be printable with inkjet. So we went on with this. We took materials that we didn't really know whether they would be printable or not. We were really interested in biodegradable, implantable materials that were 3D printable, and so we designed a material synthesis program where we created these oligomers that were biodegradable, and we coupled them with various different solvents, in this case PEGDA and NVP, again, blended them and asked the question, are they printable or not? And a priori, we didn't know that, and after our printability assessment, we were able to identify mixtures of these materials that would eventually end up to be printable. So I don't really want to say very much about geometry because it's well known that geometry affects the properties, and there's a lot of studies out in the literature. But just to add up our tuppence on this, we used to begin with, we used a very simple extrusion method to demonstrate that we could get tablets that used a single formulation. And this is important if you're, say, using a pharmacy, developing a pharmacy in a hospital where you don't want people to be making up formulations every single time they want to make a tablet. You just want the formulation there, and it just needs to meet the demand out of the analysis. And so here we work with GSK to show that a single formulation could produce an instant release, which is with the mesh tablet, or a more sustained release, which is a solid tablet. And these met GSK requirements in their QTPP. So we're really confident that this could satisfy production requirements. We also did this with inkjet, and we backed that with modeling. Modeling was able to tell us what structures we should generate in order to be able to get instant release or sustained release. Just to go back again to the formulation I talked about before for poorly soluble drugs, and you may have noticed when I put that slide up, they had different shapes. So there again, we were exploring the way in which shape could affect the release profile. And so we get different release profiles for different shapes, as you'd expect. But one of the things that we wanted to note, and was interesting here, is that previously and a lot of the research in the literature has shown that you get very predictable release from different geometries, principally because it can be aligned to the surface to volume area and the diffusion path length. Here, actually, we didn't get that, and you don't always get that because this is a swellable formulation and it's also an erodible formulation, and that does not necessarily align exactly to surface to volume calculations you might get out of a geometry. So you have to be a little bit careful when you're trying to get predictive quantities out of that kind of calculation. So some of the sort of more complex things that we've been able to uncover is the role of microstructure in the release of drugs in 3D printed formulations. Obviously, microstructure and formulation is well known in controlling drug release in solid dosage forms and other drug delivery systems. But here it's relatively unexplored in 3D printing. And so in our theme of trying to find rapid ways in which we can uncover what's going on in our systems in materials, we took our biodegradable solvent mixtures that we were screening for printability and decided to ask the question, if we understood the microstructure at a single droplet level, does that give us a guide to how it's going to perform when we start actually fabricating with these systems? So what we did was we performed a screen with various different combinations of our materials. So for example, on the top left, we've got polycaprolactone mixed with PEGDA, or we've got an MVP and various different other core materials, polylactic acid, methylene carbonate, and ask the question, what kind of microstructures should we expect from that? And then we decided to back that with an analysis based on the Flurry-Huggins interaction parameter, which gave us a really nice correlation between the kind of observations that we got and the interaction parameter values that we found. And we found that we could classify all our microstructures according to the Flurry-Huggins interaction parameter, ranging from homogeneous to dispersed material to complete separation in a core shell type of environment. And then when we printed it and looked at the microstructure with Toff-Sims microstructures and aligned that to the kind of behaviors that we're getting when we did dissolution, we found that we saw the same kinds of microstructures in 3D printed structures that we saw in a single drop, and we're able to relate essentially this screen as a predictive element or a predictive guide to what we should expect from a 3D printed, or at least an inkjet printed structure. A different way of controlling the microstructure we found was to actually covalently bond the drug to the polymer backbone, and this gave us a really strong, intimate and exquisite way of controlling the release, and also a really nice a benefit of being able to get a really high dose of drug into our formulation without everything precipitating out and falling out. And then finally, because in 3D printing we can print more than one material at the same time, so you can have two inkjet heads, we were able to show that we can select, for example, for our bacterial resistant materials, we can choose materials that were flexible, materials that were resistant hard, or sorry, materials that were high modulus, were kind of brittle, and we can start to design materials that had structural properties by design as well as cell instructive properties by design. And in the bottom here you can see a very simple example of designing a joint, a bacterial resistant joint that has a flexible material printed in the middle and solid at the edge. So just to conclude, we have additive manufacturing, which we know is a very nice on-demand manufacturing tool. It allows us to have personalization, but those personalizations are not just about shape, they're about material composition, being able to design that in and being able to design the microstructure in. And this can all be backed by screening and understanding of how this microstructure arises. And so, yeah, this is our team. Thanks very much to all the team for their contributions. It's a real team effort. And I would gladly take any questions in the chat, or if we have time for chat in the voice.
Yeah, thank you very much, Ricky. So you've got 3 questions that have come through to you on the chat. Very quickly, we'll just take one and then we'll move on, if that's okay. So the first one, which was from Pedro, which just asked about the geometry of the nozzle, which is used in printing. Is it important? Is this another lever, or is it important?
Right, so in some forms of 3D printing, the shape of the nozzle is really important. So with extrusion-based printing, the shape of the nozzle can clearly have a really strong influence on the kind of bead that you get. We found in inkjet printing, the shape of the nozzle isn't really that important. So the nozzle that I showed, the inkjet printing nozzle that I showed, is actually a square nozzle, but you can get circular nozzles as well. But essentially the distance between the nozzle outlet and the substrate is sufficiently far that the drop forms a spherical drop, or something near to a spherical drop, and that shape is then independent of the shape of the nozzle plate.
Brilliant. Thank you very much. Well, Pedro, hope that answered your question. If not, please feel free to follow up. Ricky, if you could just follow up with the other questions in the chat, please, that would be fantastic. Yeah. Thank you. And everybody else, please feel free to add additional questions for anybody, for either Derek or Ricky or anybody from this morning as well.